I think we can start now. Okay. Good evening, everybody. Uh, my name is Shomo. I am a third year PhD student at IISc, and I am the PMRF TA for this course. So, I don't know how many of you have joined sessions like this before, so I'll just mention what I do in the sessions. And yeah, so basically every week we'll have this 2 hour sessions on Tuesdays, this time 6 to 8 p.m. So it's a 2 hour session. What I do generally is discuss that week's content of your course uh, for 1 to 1.5 hours, and then I discuss assignments, the previous assignments, not your current assignments, and like we do it in a interactive manner. We discuss the answers and questions of those assignments. Uh, yeah, so about me, I am currently in my third year. My expertise primarily is with genomics, but although I have covered other omics like proteomics and transcriptomics and other omics technologies as well, but my primary field of interest is genomics. I work in genomics. So what happens is like in the course a lot of topics are discussed. So the topics that I particularly have expertise in, I try to dig a little bit deeper. So primarily the genomics part of this course, I'll dig a little bit deeper on that and like maybe show you a little bit on how we generate, not generating the data, but after generating the data, how the data looks like and how we process that data across different pipelines and different algorithms. So before I start today's session, a little bit of I would like to know the little bit of background of all of you. So can you just like unmute yourselves and like let me know what is your background, like what you have studied? Are you a student or are you a professional or anything else? Or you can write in the chat as well. So I'll have a idea about what is the demography of the people that I am teaching. So it'll be helpful for me and also helpful for you. I'll design the lectures like that. So yeah, you can unmute yourself and let me know, or you can write in the chat, whichever way you are comfortable in. Yeah, go on.
Hello all, my name is Hariharan. I am from Bangalore.
Okay.
I am a final year MSc Life Sciences student from
Okay.
Bangalore University.
Okay.
My expertise is microbiology.
Okay. Okay. That's great. Others? Okay. I can see Avnish has replied. BSc second year. Okay.
Hello,
Yeah.
good evening, Mr. Majumdar. I am Dr. Priyanka Dhar from Kolkata.
Okay.
I am an assistant professor in botany
Okay.
in Brahmananda Kshetrendra College. My
Okay.
PhD was in life science, and currently I am teaching in the plant biotechnology and other aspects of plant science.
Okay. Okay. That's great. That's great. Okay. I can see some other chats as well. Okay. BSc, MSc. Okay. I can see a PhD here. BTech. Okay. 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 So there is a like a mix of different levels here. I can see BSc students, PhD students, even assistant professors. Okay, that's great. That's great. Thank you. I'll now get started with the session. I hope you can see my screen. Sorry. I hope you can see my screen. Okay. So welcome to week 1 of advances in omics. So in this session primarily we'll be discussing about some older sequencing techniques and basically like how we have evolved to current sequencing techniques like next generation sequencing from Illumina, or even now we have long read sequencing, ONT and PacBio and so on and so forth. But this session will mostly be focusing on some older techniques, some of which we still use, for example Sanger, is still used in certain cases, but some others like Maxim Gilbert and all those things probably are not so much in use in today's world. We have better and more efficient sequencing algorithms, but we still have to study about them to know the concept of how and why we do sequencing, right? So let's start with the first technology. I think this is the oldest one, but there is some debate about this and the Sanger, but technically this is the oldest sequencing technique. This was first established in 1977 by two scientists called Alan Maxim and Walter Gilbert. So this technique uses chemical modification of DNA, and the enzymes identify these chemical modifications on the DNA, and they subsequently break the bonds, the phosphodiester bonds across the DNA backbone, and then then you purify the samples of these double-stranded DNA, and then you do radioactive labeling and basically try to find out what base is at that position at which the cleavage has occurred. So basically what happens is you take these DNA strands, and then you radioactively label at one of the 5' ends of the DNA, and then you do chemical treatment. As I said, you use enzymes which identify these modifications on the DNA, and they break it down into small proportions of 1, 2, or 4 nucleotide bases at each of those four reactions. It can be G, A plus G, C, and C plus T. And the concentration of these modifying chemicals is controlled to introduce on average 1 modification per DNA base molecule, and the modification point of the DNA base molecule is basically the cut site at which the enzymes will cut the DNA, right? So first, how you do it is, first you separate the 2 strands of DNA, then you label the DNA with respect to radioactive, basically radioactively label the DNA, and you give different, and you put it in different test tubes of 4 samples. So in each tube it is subjected with chemical treatment that specifically destroys 1 or 2 bases. So it can be a G or a C, or it can be a plus G or T plus C. It identifies that, and it cuts the DNA at those locations. So the DNA is digested at in the 4 samples at the sites of G, A plus G, T plus C, and C, and this results in the formation of a series of labeled fragments of different lengths, right? So similar kind of technique is also used in Sanger, where the different lengths are also estimated. How do you know the DNA has been cut at different lengths? What you do is you do some sort of a electrophoresis assay, and the amount of DNA that is traveled in that assay, in that gel, will tell you what is the weight of the DNA molecule. So the higher the weight, the less it will travel. The lesser the weight, the further it will travel. I hope you all know a little bit about gel electrophoresis. So what happens is the actual length of the fragment on the site at which the base is destroyed from the labeled end, so that fragment is taken and measured. And thus, for instance, if there are C residues at position 4, 7, and 10 away from the labeled end, then the treatment of the DNA that specifically destroys the C will give you labeled pieces of length 3, 6, and 9. So you had the C base at the 4th position, the 7th position, and the 10th position, and when after modification and after the cut is happened, you will get sequence length of 3, 6, and 9, because at the 4th, 7th, and 10th position the C was there. Then the labeled DNA fragments are obtained across 4 tubes and are subject to gel electrophoresis side by side, and they are detected by autoradiograph. And the sequences of the bases in the DNA can be constructed from the bands on the electrophoresis. As I said, according just a minute. Yeah. As I said, according to the molecular weight or the length of which is also proportional to the length of the DNA fragment, the distance covered in the gel will determine what is the weight of the DNA sample, right? So after the cleavage, again it is put through the gel electrophoresis, and across and depending on depending on how much the DNA has traveled, you can determine the length of the DNA. And say, for example, you have two fragments, as I said, like if you had fourth and the seventh position, you have two fragments of length 3 and 6. So the 6 base pair DNA will travel lesser, and the 3 base pair DNA will travel farther. And you'll know that this is a DNA of length 3 base pairs, and this is a DNA of length 6 base pairs, and based on whichever enzymes has cut the DNA, you will know that what position, what nucleotide is present at that position. So the idea is simple, that you take the DNA, you modify the bases, you cut the DNA according to the modification of the bases, and then you run it on a gel electrophoresis, and then according to the length of the DNA, it will travel farther or lesser, and according to which enzyme you have used, you will know that at that position which is the DNA base that is present, right? So the principle is very similar to what is used in Sanger as well, right? So okay, before we go to Sanger, this is basically a representation of how Maxim Gilbert method works. So you take the double-stranded DNA, make it single-stranded, distribute it into 4 tubes, and it is using enzymes. Specific bases will be destroyed and fragments will be formed, and then you separate these fragments by gel electrophoresis. So these are the 4 tubes from which this has come. So for example, so the farther it goes, as I said, the farther it goes, the shorter in length and the lesser in molecular weight it is. So this is the shortest length, where you have a T plus C, and the next one is probably a C, then the next one will probably a, then A, and then a couple of G's, and so on and so forth. So from the gel electrophoresis only, you can just work back on what the nucleotide sequence was based on the on these parallel radiographs that you generate using this different four different samples, right? So as I was saying, the idea is very similar to Sanger sequencing, but in Sanger sequencing there's a small caveat, and the small important thing that is still followed till date in also techniques like Illumina sequencing, which is known as sequencing by synthesis. So basically what you do is you determine the sequence by synthesizing the sequence, right? So how do you do it? So first let me talk a little bit about the Sanger sequencing. So basically Frederick Sanger won Nobel Prize for this technique in 1977, and this was one of the very few techniques that actually held the it passed the test of time, and it is still used in different contexts. But the main idea of Sanger sequencing is that concept of sequencing by synthesis, as I was saying. So in this case, you have DNA polymerases, which uses naturally occurring DNTPs in the solution to make the complementary strand of DNA. We'll come to that, how it is done later, but this is the main differentiating factor of Sanger sequencing with Maxim Gilbert or other older techniques, is it used sequencing by synthesis, right? So what does it mean, sequencing by synthesis? Is basically when you have a double-stranded DNA, you break it down into single strand, and then you use DNA polymerase, and you use the naturally occurring DNTPs in the solution. So when you are sequencing, you divide the double-stranded DNA into single strand, you put it in the solution, and in that solution there will be other DNTPs present. So D, ATP, D, CTP, TTP, and so on and so forth. So right, so those DNTPs that are present freely in the solution can be used by the DNA polymerase to create this new strand of DNA, right? So the idea is that also if you start sequencing, if you start adding these bases using DNA polymerase, there is no way to generally stop that. It will only stop when it has reached the end, and there is the termination factor, and then it will stop the synthesis. But in the sequencing case, in the Sanger sequencing case, what they do is they use they figured out a way to terminate synthesis using a modified version of these DNTPs. So what happens is you have a single strand of DNA, say ATGC. So DNA polymerase comes and bind. For A, it takes a T. For T, it takes a A. For G, it takes a C, and so on and so forth. And when it takes, say for example, there is a DDNTP of A in there. So when there is a T, what will happen is the DNA polymerase will add a DDNTP of complementary to A, which will be DTTP, and after that the sequence, the synthesis will not occur, because instead of DNTP you have added DDNTP, right? So what happens is, like, by the use of these DDNTPs, you can stop the synthesis at the required base that you want. So as I was saying, DNA polymerase can attach a DDNTP to its sequence like with other DNTPs, but it cannot attach anything to the DDNTP. So the point at which the DDNTP has been attached, there the sequencing, the synthesis will stop, right? So it will halt the replication of the DNA molecule. So this is a representation about how the Sanger sequencing occurs. So for example, you have a stretch of these single-stranded DNA. First you have to make the single-stranded from double-stranded, that you can do. Then what you do is you have a solution of DDNTPs, but in there you have a solution of DNTPs, but there you add a small fraction of these DDNTPs. So out of all these DNTPs, there are 2 DDNTPs added of T. So generally the proportion would be something like this, where you have DNTP around, if it is 99, then the DDNTP will be 1. So it will be 1 in 100 bases you will add a DDNTP. Then you also add the DNA polymerase enzymes for the synthesis. And also, if you remember DNA replication, DNA polymerase cannot start the replication by its own, right? It needs a primer. So you also have to add a little bit of a primer after which the DNA polymerase will take the synthesis forward, right? So after adding the primer, after adding the DNA polymerase will start to synthesize the rest of the DNA, and at the position it adds a DDNTP, it will stop the synthesis, because after that you cannot add a DNTP. So what will happen is, if you specify which DDNTP is added in that solution, the synthesis will stop at that particular base location, right? So for example, here you have added the DDNTP of T. So what will happen is, it will stop the synthesis at a A. So the complementary of A will be T, and instead of T, a T prime or a DDNTP of T will be added, and it will stop the synthesis. So wherever there will be T's, generally wherever there will be T's in these in this solution, it will wherever there will be A in this solution, it will add a DDNTP and it will stop the synthesis. But the proportion, the concentration of these will be much lower than the actual concentration of the whole synthesized DNA, but that is there. But still you will have these small portions of this DNA where the synthesis has stopped at a particular base that you intended. Now what you do, as I was saying, that you take these DNA elements out of the solution, and then you run it on a gel electrophoresis. So in this case, as I was saying, that you have added T DDNTPs, so the synthesis will stop at A. Just a second. Yeah, so you get this fragmented portions, fragmented DNA sequences, which you can run through in gel electrophoresis. So what you can do is, you can run all of these as we were seeing here as well. You can run these in this gel electrophoresis, and based on where it stops, it will tell you what is the proportional length of that DNA fragment, right? So if a fragment stops just above another fragment, and both of them have stopped at A or T, whatever DDNTP you are using, you would know that first there was a A, then there was a T, or there is a 2 consecutive A's together there, right? So the general workflow would be to first use a PCR technique. You do you increase the amount of DNA by using PCR, and then you break the two strands into and make it into single strand, and each of the single strand will be used as a template for the DNA polymerase to work on. Then you add a template of the DNA and basically DNTPs with a few modified DDNTPs, and keep this in mind, in one solution the type of DDNTP added has to be singular. Like you cannot add a DDNTP of A, a DDNTP of T, and so on all in one sample, because then you will not know at what base the synthesis has stopped, right? So only one type of DDNTP will be used, and you add a primer for DNA polymerase to start the replication. Then you filter out sequences that end in DDNTPs, basically these ones where the synthesis has stopped, and then you put this this fragmented DNA into a gel electrophoresis, and you can isolate these strands based on the molecular weight or the length of the DNA, right? So it works pretty well. Actually, Sanger works pretty well for a short sequence of DNA. But generally what we have seen is that if you go above 700 base pairs, it is not been able to distinguish between lengths of 700 and more. So for example, so if you have a base of length say 5 versus a DNA of length 6, it can distinguish between 5 and 6 very well. But when the n increases to about 700 and so, it cannot distinguish it very efficiently. So what happens is, if you have larger fragments of DNA, the error increases exponentially. So generally Sanger sequencing is done on smaller lengths of DNA because larger lengths is not very compatible with that. So this is how the gel electrophoresis looks like, where you have with respect to Sanger sequencing, right? So you see these dark bands. These dark bands are actually DNA fragments, and based on that length, so basically the ones that have traveled the most, they will be of the shortest length, and the ones that have traveled the less will be of the highest length. And using the sequence of these lengths, you can find out what is the actual sequence of the DNA strand that you are sequencing, right? So that was Sanger. Sanger is a very well established method, as I said, which is still used to date. By the way, if you have any questions, feel free to stop me and ask. I do not mind that, but please ask questions if you do not understand. Okay. So the next type of sequencing technique that we'll be talking about is basically, the technique is called pyrosequencing, and it was developed by this company called 454 Life Sciences, which is later acquired right now by Roche Diagnostics. This technique is a little bit dated in terms of it is not used so much in the current scenario, but still it's a very interesting technique that we see. So what it does is it uses a specific enzyme which is called luciferase, and it uses luciferase to provide a signal in terms of so for example, when you have that luciferase signal from your sample, you know a particular base has been added to that particular location, and you know what base has been added. So when you do that continuously, you can generate the whole sequence, right? So generally how it works is, here you have a single stranded DNA on which you are trying sequencing by synthesis. Here you are doing sequencing by synthesis. You are having a sequence, single stranded DNA, you are adding DNTPs and DNA polymerases, and you're just creating the complementary strand. So this is basically the globular structure is the DNA polymerase, and this is the sequence that it is adding. So what happens is, when you add one of these bases, one of these DNTPs, a pyrophosphate is released, and what this pyrophosphate does is, in the solution there is AMP present, which converts it to ATP, and there is also this particular enzyme present, which is D-luciferin, and which reacts with this ATP to give luciferase, which is actually the enzyme that is formed, and it also produces a light signal. So if you have a kind of a receiver, you can detect this signal, and you can say that there is this particular base in this particular position. So wherever you have, so because you're again in a single sample, you're using a single type of DNTP, sorry, DNTP. I apologize. So in a single sample, you are using a single type of DNTP, and where you are getting this light, you know that that particular base has been added at that particular location. So you have these kind of peaks at different locations, and you know according to this peak which base is present at that location. So again, the concept is very similar. Here also you're doing sequencing by synthesis. You have a single stranded DNA, you're adding bases. Here you're not, the difference with Sanger is in Sanger you are stopping the synthesis at a particular base location. Here what you're doing is you're not stopping the synthesis, you're keeping on adding molecules, but at particular DNTPs you get this light signal, and you know the location of that DNTP, and you know what particular base has been added. But to keep in mind that you are adding the complementary base. In the previous case also, in Sanger also, you are adding the complementary base. So if the termination happens at T, that means your base at that location is A. So keep that in mind. So if a T has been added here, it means you're getting the complementary base as T and the original base as A. So generally when sequencing, we get the complementary sequence and then we convert it back to the original sequence, right? So this is the basic concept of pyrosequencing, which is kind of loosely based on Sanger, but it's not exactly Sanger, and it is actually more efficient in Sanger in terms of getting larger lengths of DNA compared to Sanger, right? Now I will talk a little bit about Illumina sequence. I have not got any slides for Illumina sequencing in this particular session. I will talk in detail about Illumina sequencing, nanopore sequencing, and other type of genomic sequencing in later sessions. But here I just want to highlight a few things about, like, you are studying about how the data is generated. So these are techniques by how the data is generated by pyrosequencing or by Sanger or Illumina, as I would discuss now. But the idea is that once you get the sequence data, what do you do with the data? What kind of data does it generate? But before that, I think I would like to, I don't know if I can show you, but I certainly would like you to see this particular one second. There is a Illumina sequencing. Yeah, so this is the video actually. So I don't know if you can hear, if you'll be able to hear this voiceover or not, because that actually kind of explains it. If not, I'll just send this video link in the description. One second. Okay. Yeah. So I'd like to just, this is a 5-minute video. I don't think it'll take much time to explain this, but like still I would want you to actually take a look at the video, or also maybe take a look from here.
The Illumina sequencing workflow is composed of 4 basic steps: sample prep, cluster generation, sequencing.
This sequencing is again by sequencing by synthesis. You are adding a base pair and you're going to know what the base pair has been added there, right?
And data analysis. There are a number of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification,
so these kind of details of why you are adding adapters, how much of the adapter length should be added, and those are technical details that you, for now, you can ignore, but focus on the main concept of the synthesis part, like how the synthesis is occurring and how the data is getting generated, right? These technicalities you can like forget for now. We can discuss later, but yeah. focus
Additional
on the-
motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
Yeah, so this is how actually what the flow cell looks like, right? So here you have different a lot of flow cells in this small structure, but this is how the chip looks like, and inside each of these lanes you have these different regions where these DNA strands can go and bind, and then sequencing by synthesis can occur.
Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn, composed of two types of oligos.
So inside each lane you have these small oligonucleotides, which is basically complementary to the adapter that you added in the previous step. It was showing and you were adding adapters. So these oligos will be complementary to the adapters. So when you put that DNA inside these lanes, what it will do is the oligo will come and bind to the adapter, and it will form half double-stranded DNA with just the adapter and the oligos like fused together, and then the rest of the complementary strand will be sequenced by DNA polymerase.
Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process,
Bridge amplification and clonal amplification, these are things specific to Illumina. These are just to increase the throughput, the amount of DNA that you are synthesizing, but these do not primarily affect the actual concept of the sequencing, right? So these are techniques used to amplify the DNA. These are techniques used to get higher throughput and so on, but like these are not actually core to the actual concept of the sequencing.
the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge.
So this bridge-like structure, so formation of this bridge-like structure is also known as bridge amplification. It's as I was saying, it's just a process of amplifying the amount of DNA, increasing the throughput. That's all.
This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming.
Now actually the sequencing occurs, which is the sequencing by synthesis.
Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete.
So what is the difference of Illumina with Sanger? In Sanger you used DDNTPs, right? So whenever you added a DDNTP, it stopped the synthesis. But in Illumina, what they do is they do not stop the synthesis completely, like in pyrosequencing also. What they do is they use a marker of some sort. So when a particular base is added, a particular signal will be emitted, which you can capture and know that this position has this particular signal, which is corresponding to this particular base. So in this case, fluorescently tagged DNTPs are added. So each DNTP, when added to the sequence, it emits a particular light, particular signal, which is captured and which is then converted to a base by, there are other softwares actually that look in that kind of read these signals and they convert it into base calls. So those are called as base calling softwares. Just a second.
For addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process.
This is why it is called high throughput, because you can sequence millions of these reads together at one go, right? Which was not possible in Sanger. Right? Sanger, you had only 4 tubes and one DNA strand, and you put it in 4 tubes and see where the sequence is terminating, and then you run it through gel electrophoresis, and then you just get the sequence of one strand, one small portion of the DNA. Here what is happening is, due to these flow cells and these lanes inside which you have these millions of copies of these DNA reads, all of these, not all, but a lot of these DNA fragments are sequenced simultaneously. So that is where the concept of high throughput comes, that you can sequence a lot of DNA together, right? You do not have to do it sequentially.
This image
So this is what as I was saying, that there will be a light source which will be emitted, and there will be a signal receiver which will receive the signal, and then it will call a particular base on that signal, right? So this is actually the image that is produced after sequencing. This is actually the output of the sequencing. You do not get bases as output of the sequencing. This is the output of the sequencing, this image file. This will go through a particular base calling software which will identify these emitted signals at different positions, and it will call that with certain amount of probability, because this is always going to be, there will be some errors associated with it. So with a certain amount of probability, what is the base at that position. So this is actually the output of the sequencing, not as as the reads that you would hope, right? There will be another software which will look at this image and tell you what bases are there.
represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1.
So these indexes are generated to kind of tell you which read has this particular sequence. So it's indexing of the read is just tagging the read with a particular, say for example a barcode, to tell you, if you know the index of the read, you know the whole read. So that is why these indices are generated.
Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand.
Here. So in the first step you had cleaved off the reverse strand and only the forward strand was remaining, and you sequence the forward strand. In the second step you are removing the forward strand and sequencing the reverse strand. So in Illumina what is happening is you are sequencing the reads twice. A single read will be sequenced twice, one from 5' to 3', another from 3' to 5'. So generally you have two sequences, one for the forward read, one for the reverse read. So this increases the accuracy of the technique, and also it helps in further downstream things like variant calling and detection of larger variants in the DNA and so on and so forth. We'll come to that later, but generally you get a forward read and a reverse read with Illumina.
Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired
process.
read length is achieved. The Read 2 product is then washed away. This entire process generates millions of
Okay, we'll not go into the data analysis part here. We'll discuss that in great detail in later sessions. But yeah, I hope you got an idea about what is sequencing by synthesis and how does it occur. So Illumina is basically, which is the next generation sequencing technique. This has been here for like close to more than 10 years now, more than like probably 15 years now. Illumina is here, and this Illumina sequencing is very much used till date. Every large scale genomic studies that you see is done using sequencing of Illumina, and the only drawback of Illumina sequencing is basically it produces again shorter reads. As I said, in Sanger also the issue is that the larger the read becomes, it becomes harder to differentiate between what is the sequence at position L versus position L+1. So the resolution decreases as the length of the read increases. Here also the issue is that Illumina can only sequence up to 500 base pairs or 700 base pairs maximum, right? It cannot sequence beyond that. Beyond that, it is you have to look at further newer generation technologies like of long read sequencing, like Oxford Nanopore, like your PacBio and so on. So there they can generate this high length reads which can be go up to like 50 kb, and even in some cases in MBs also it can go up, right? So those will be very high length reads, but in Illumina generally this is around 500 to 700 base pair. So Illumina is called as short read sequencing because we have techniques now which can generate such higher longer reads, right? So but still Illumina is a very well used technique till date. We in our lab also use Illumina sequencing to sequence human samples as well. So it's a very well established method, and this is basically the high throughput sequencing, the next generation sequencing. The first generation would be your Sanger and your Maxim Gilbert and so on, right? But you see the Sanger concepts are still applied to this next generation sequencing. The Sanger concept was that you do sequencing by synthesizing the DNA strand. Here also you are doing sequencing by synthesizing a DNA strand. How you are controlling the length of the sequence that you are synthesizing, that is highly different between Illumina and your older Sanger techniques. And also what length and also like how many of these reads can you sequence together, that is also vastly different between Illumina, where you can have millions of these reads sequenced together, which we call as high throughput, and in Sanger it is not possible, right? Okay, coming back, so I hope you have got an idea about like these things are actually covered in detail in your course, but I just wanted to give you like an overview idea about how these sequencing techniques are used, where it is used, and how the data actually looks like. So when you have a sequencing data, what do you get, right? So I'll just quickly go through this particular set of slides, and then we'll move on to the questions, assignment questions, right? So generally the output of these next generation sequencing or like Illumina or whatever you're doing are generally FASTQ files. So I think a lot of you might have heard about what FASTQ files are. So basically these are files that are containing the sequence of the DNA. How it looks is basically this. So you have a particular line telling you about the read. So there are four parts of this file. So first will be an identifier line. This will be the identifier line. This will be next will be the sequence. So this is the sequence. Then you have a line which almost simply puts a plus sign, which can be optionally followed by a repeat of the ID line. And the next line is the important one. This is basically ASCII coded quality parameters of this particular basis. So for example, for T, the ASCII coded quality value is A. So if you convert it into numerical value, it will come as some numerical value, but what the quality parameter is telling you is what is the proportion, what is the confidence that you have that particular base in that location, right? So what generally the values are would be, say for example, you assume that there is an error in one base if you sequence, say for example, 1000 bases. So 1 in 1000 you have an error rate, right? So that would be 10 to the power -3. The quality parameter is generally called Fred scale, where you have like negative log 10 log 10 of your error rate. So in this case it will be negative 10 log 10 of 10 to the power -3. So if you just resolve this, it will come as 30. So if your quality parameter is 30, that means that the error rate of your experiment or of your sequencer is 1 in 1000. So like that, a quality parameter is assigned across all the bases, and it tells you how confident the base calling software is in terms of saying that this base is present in this particular position. So this is how the output, a little bit of processed output of the sequencing experiment would look like. So you have an identifier line, then you have a sequence, then you have the quality parameters of that sequence, right? So generally, after we get this data, we do a little bit of quality control and we check certain things. Say for example, if there is any contaminated bases present. So as you were seeing in Illumina, when you are doing the sequencing, you are adding adapter sequences, you are adding other sequences which will help and go bind to the oligomers in the, oligos in basically the flow cells. So these extra bits of DNA that you have added, those can come as contamination as well, right? So identifying if there is any contamination, identifying if any other molecule other than DNA might have also been sequenced together, for example an rRNA or some other type of RNA might have been sequenced together with your DNA. You would need to remove those sequences from your data. And also as a validation check, there is like a fixed amount of GC content that is present with in a particular organism. So if you know that, you can also calculate what is the proportion of GC content in your data and kind of validate, okay, this data is good because my GC content actually matches. Just a second. This, let me see. Okay, yeah, the quality parameters, as I was saying, it is based on the, so just let me go back here.
Reads.
So as I was saying, this is the actual output of the sequencer, right? So this is basically all the signals, fluorescent signals that has been captured during the sequencing experiment. Now this data will be input to a base calling software which will look at this particular set of images and tell you that what base is there at what position, right? But the base calling software is a prediction model. Generally, the current base calling softwares will use some sort of neural network models, some sort of highly like high level deep learning models to tell you that this signal corresponds to this base, right? So there will always be an error associated with it, be it at the sequencing level where the light is getting generated, or at the base calling level where the base caller is converting this light signal into the base, what base is there or not. So this error is quantified in terms of the quality parameter. So if your error rate is, as I was saying, 1 in 1000, right? So 1 in 1000 meaning 1 by 1000, which is 10 to the power negative 3. The quality parameter is given as a Fred scale. The Fred scale is a particular type of scale that is used across these omics platforms to tell you about the quality of that particular location, be it a base, be it can be an amino acid if you are sequencing, say for example proteins, or if you are sequencing RNA, what is the base there. And at every, when you're sequencing, at every position there will be a quality parameter associated with it which will tell you what is the error rate, right? So if it is 1 in 1000, it will be 10 to the power negative 3. Yeah, it will be 10 to the power negative 3. And if you convert it to Fred scale is basically negative 10 log your error rate, right? So your error rate will be 10 to the power minus 3. So negative log of 10 to the power minus 3 will be 3, and you also have a 10 in front of that, so 30. So if your quality parameter is 30, it means your error rate is 1 in 1000. So higher the quality parameter goes, the error rate decreases. So if, for example, if it is 60, if your quality parameter is 60, then your error rate is 1 in every 10 to the power 6 bases. So that is very good, right? So quality parameter greater than 50 and 60 is very, very good. We generally take 30 is okay with us, right? If you go around 30, that is fine. So that is how the quality parameter is calculated. And it is not only in terms of sequencing. After sequencing, when you use this data to see if there are any mutations in the gene or not, in a genome or not, you are going to use a particular software to tell you, okay, that there is a mismatch in the reads in this location, so there might be a mutation here. So that also comes with a quality parameter, because all of these are predictions. All of these are predictions, and there will always be an error rate associated with predictions, and quality parameter will tell you basically what is the error rate. I hope that is clear. Just a second. Okay. Okay, as I was saying, after you get the data, you would like to look at the quality of the data. Is the sequencing done all right? If there was any contamination or not, and so on and so forth. So there are different tools to look into it. So one of them is FastQC. So what it happens is it's a very well-known tool. You can just look up, I think, in Google, and you get it. It's one of the oldest ones. FastQC. Yeah, it's a quality control tool. This is a Java based tool. It's very old, but yeah, if you have any interest, you can definitely take a look. Okay, so what it does is it takes the FASTQ file, and then it creates different figures and plots which will tell you what kind of data you are looking at. So for example, it will first tell you what are the total number of sequences, what is the average sequence length, what is the GC content, right? Then you can also look in detail what is the base quality. As I was saying, there is a quality associated with each base, right? So as the length of the read increases, so as the position in the read increases, generally the quality decreases. So it's a decreasing function, and this function is kind of acceleratedly decreasing. So if it is so if you there's also a question in the assignment also I saw related to this. So for example, if you are at the initial basis, it will have quite higher quality, but as you increase the length of the sequence, the quality falls. So here the curve, if you consider this curve, right, this curve will be decreasing, and the rate of decrease of this curve will also increase. So basically both your first and second differential of this curve will be negative, because both are increasing. So basically your rate of decrease is increasing, and also it is decreasing, right? So you also get an idea about the base quality. You also get, in terms of per sequence, what is the quality score. These are again quality parameters. And also GC content. So it gives you a theoretical distribution of, say for example, if you're looking at human sequences, the theoretical distribution would be something like this, which is on the x-axis you have the mean GC content, and on the y-axis you have the sequence lengths. So basically this is how it should look like, and the red one is basically how your GC content per read actually looks like. So if there is a very high difference between your theoretical and your observed count distributions, you would think that there has been some contamination or something is wrong with your sequencing data, right? Then you also look at you can also look at overrepresented sequence. So for example, one particular sequence that has come up multiple times in your data, right? So for example, here these sequences have come up 2500, around 2500, around 2000 times in your data set. So in most cases what can happen is these overrepresented sequence can be contaminations. They can be vector sequences or adapter sequences, as I was saying. So if you have these overrepresented, very high amount of these overrepresented sequences, the idea is that you remove this before you further process your data. So okay, so we'll not cover about how right now about what you do after you check all the quality parameters. Generally you do alignment with a known reference genome, then you do variant calling on that, and then you find what the mutations are, and so on and so forth. We are not going to all those details today, but generally this is how a quality control of sequencing data looks like. So basically you look at quality of the bases that has been sequenced, you look at the GC content, and you also look at overrepresented sequences in terms of like if there is contamination or not. So yeah, that would conclude all I had to discuss in terms of the content. That as I said during the start of the session as well, the content that I will be discussing will not exactly 100% overlap with the content that sir has discussed in his in your like weekly videos. What I will try to do is give you a larger overview of like where and how these technologies are used and what you can do with these things and so on. In your regular videos of sir, you'll get to know more about the concepts and more theoretical, a little bit more theoretical. I would like to do it a little bit more not practical. I cannot do practical here, but like a little bit of idea about how practically these technologies are used. So yeah, so that is all in terms of the discussion I wanted to have. Next I will go into the different assignment problems. But before that, if you have any questions. Yeah, so this is a very interesting question, that why the base quality scores will decrease. It's a property of sequencing by synthesis only, right? So as I was saying that in Sanger, what happens is when you increase the length of the sequence, the resolution drops, right? So the more you sequence, the less confident you are that there is a correct base at that you know the correct base at that location, right? So this is a general drawback of sequencing by synthesis, is that the larger the length of the read, the lower the quality you will have after a certain point. So since in Illumina also we are doing sequencing by synthesis, here also that concept follows, that after the signals get accumulated, after like you have a 500, say for example 1,000 base pair read, you start from the first base, and then you are adding up these signals, not adding up, like you are generating these signals, and then you are getting these signals and you're keeping it. And when you are reaching, say for example 600 or 700 base pairs, what happens is the resolution of the signals decrease, because you are doing sequencing by synthesis. You have gotten so many signals, it is going to be very hard for you to distinguish. The resolution, as I said, will go down. So the quality of the bases at the lower the higher end of these reads, at the last portion of these reads, will be lower. But one way to mitigate that would be the reverse sequencing that I showed in Illumina as well. So you have a forward strand, also you have a reverse strand of the same DNA sequence. So you are sequencing the DNA. So for example, if you had 1,000 base pair DNA, you're sequencing the DNA first one from base 1 to base 1,000, and then base 1,000 to base 1. So if you do that diligently, this thing might get mitigated, but still you can have issues. And practically also we see issues in terms of the quality falling off with the increase of the length of the read. So what happens is in current or in long read sequencing, the current like state of the art technology, you can say, where you can have this large, very high length of these DNA fragments, 10 kb, 20 kb, 50 kb, and so on. What happens is they do not use sequencing by synthesis, because sequencing by synthesis will not work when the DNA fragments are so large. It is going to be very hard for you to distinguish these sequences, these bases, as the length increases. So there they use different techniques. So for example, in nanopore technology, what they use is there's a I hope all of you are familiar with the, what is the name, F0F1 ATP synthase, the hydrogen pump actually. So there are two motor proteins, right? So there is a motor protein on top, there's a motor protein below, and they kind of move relative to one another. So in Oxford nanopore technology, what they use is they use a similar protein like that, and they pass the DNA fragment through that protein, and also they pass current through that protein, and there the change in current will tell you what base is there at what location. So when you're doing this larger length DNA sequencing, you cannot use sequencing by synthesis. There you have to use different techniques, like Oxford nanopore uses current, PacBio uses some other hi-fi reads technology, and so on and so forth. So generally that is the drawback, that if you use sequencing by synthesis. Mm, no, technically, as I was saying, like actually depends. I think I do not prefer calling it by second generation, third generation. I like to call the name of the technology itself, because this is confusing. There is a little bit of debate of what is second gen, what is third gen. So by third gen, if you mean the current state of the art technologies like long read sequencing of Oxford Nanopore or PacBio, there they do not use sequencing by synthesis. But Illumina, if you consider that as second or third, whatever, Illumina uses sequencing by synthesis. Okay, any more questions? Or not even questions, if you want to discuss anything, if you have any thoughts, please share. We are here to discuss. Okay, yeah. Okay, let me go back here. Sorry. Yeah. Okay, so pyro sequencing along with other sequencing by synthesis technologies have some basic drawbacks. Firstly, as I was stating, the short length of the DNA. That is true for all sequencing by synthesis technologies, including Sanger, including Illumina, and so on. So here the drawback is basically you cannot have a sequence more than a certain length. The major drawback of pyro sequencing with respect to other newer technologies like Illumina is basically in Illumina, you can use millions of reads and you can sequence them together. The throughput is much higher than in pyro sequencing, because this is a sequential process. You cannot parallelize. You can parallelize this, but this is very hard to parallelize. But in Illumina, it is the whole concept is based on parallelizing thousands and thousands of reads to be generated together. So generally, Illumina, the major, as I would like to say, the advantage of Illumina over pyro sequencing is the high throughput output. But in general, in terms of the principle, the drawbacks are very similar to Illumina and Sanger, which is basically the read length. And also another thing, if you would like to discuss, I don't know, like if I should or not. So, are you aware of what are homopolymer regions? So homopolymer regions is, for example, you have GAA, then you have, say for example, five A's here. Instead of TTC, you have 5 A's. G, then followed by 5 A's. Right. So what will happen is, when you are doing this whole procedure, in your output you will have 5 continuous peaks, and in some cases what can happen is you will not be able to distinguish if there are 4 peaks, if there are 5 peaks, if there are 7 peaks or not, because the signals will be flat, because there will be no peak as per se. So because if you have the same nucleotide 5 times, the signal will be, it will go up and then it will become flat and then it will come down. There will be no peak per se if you have a homopolymer region, which is like the same base repeated like 5, 7, 8 times, whatever. So there, if you are depending on these peaks to tell you what base is there, in case of homopolymer regions, the issue that can arise is that you will know what bases are there. So if there are, like, say for example, as I was saying, 5 A's, you will know that there is an A there. but it is going to be very hard to distinguish if there are 4As or 5As or 7As or whatever. So in homopolymer regions, it becomes much more problematic. And other than that, the other drawbacks are mostly similar for all sequencing by synthesis technologies. The drawback with respect to Illumina is that Illumina can do high throughput. This cannot. Right. So I think I might have covered all the drawbacks. Yeah. So there are base calling softwares for Illumina. As I was saying, nowadays the softwares are very sophisticated in terms of they use different levels of deep learning networks. I have forgotten the names. Okay, let me see. Illumina base calling. No. So for long read and all, we work with daily. So there we use something called Dorado. I've forgotten the Illumina one. Yeah, IBIS is one of them. So this is the publication for that. So yeah, this is the name of the software. This is an older one. I think this is 2009. There has been newer softwares, but yeah, if you want to know how it is done, this paper would be, I think, enough. But as I was saying, like right now we have, I think, deep learning and based technologies. Deep learning. Yeah, so for example, this one. Yeah, so there are a lot of softwares that come out for base calling, but Illumina has its own. So I think IBIS is one of Illumina's. There are newer also. Yeah, this particular software does all: PacBio, Illumina, ONT, all of them together. So yeah, there are a lot of softwares you can find, but if you want to know how any one of them works, I think this should be enough. Any other questions, or we'll move on to the assignment questions? And guys, I will request one more thing, is basically it becomes very difficult. I have also faced this in earlier sessions also. It becomes very difficult for me to stop the lectures and then let you in. So my suggestion would be like to join at the beginning of the class only, even if you do not listen. And by the way, all of these sessions are recorded, and this will be put on YouTube, and you'll have the links. So even if you miss any one of them, it's not a problem. You'll find these recorded sessions on YouTube. And my advice would be, if you are joining this session, try to join within time, because it becomes very hard for me to kind of go back and forth from the material and then again admitting you and then again going back. So it breaks the flow. So it would be my request to like join at the beginning, or like if you don't want to join, it's fine. These will be recorded in YouTube also. But yeah, so generally what I do is I wait for 5-10 minutes at the beginning for people to join. So even if you're 5-10 minutes late, it's fine. You can join. It's okay. But like if you're joining in the very middle, it becomes very difficult for me. I basically lose my flow. Okay. Any more questions, or we'll move on to the assignment questions? Okay, then we'll move on. Okay, so how I do it is I put the question on the screen for whatever amount of time, 30 seconds to 1 minute or 2 minutes, depending on the type of the question. and then I discuss the answers. You can write the answers in the chat. I'll check the chat and then I'll discuss. Okay. I'll just turn off my camera because I'm having some internet issues. So probably this will be better. Yeah. Okay. Please write your answers in the chat. Today's like week 1 questions will be very introductory, very easy for a lot of you, but still. Uh, yeah, these questions shouldn't take much of time. Yeah. Thank you, Parul. Okay. Yeah, it's all of the above. Omics is basically a field which encompasses all of these things, and basically what it tries to do is quantification of different biological modules is how I try to best remember it. Is basically if you want to quantify what is the amount of DNA or what type of DNA is there, you do DNA sequencing. If you want to quantify what type of RNA and what gene expression is happening, you do transcriptomics. What kind of methylation patterns are there on the DNA, you do epigenomics, and so on and so forth. So basically it is a quantification of different biological modules. uh, Whatever module you want to quantify, you use that type of omics. So okay, I hope this was okay. Moving on. This is also very easy. Yeah, it's correct. Yeah, it's first generation. It's one of the like oldest. Okay, moving on. These kind of questions, the thing is, if you do not know the answer, you will not be able to answer. So it's fine. Like if you do not know the answer, it's okay. But definitely, I would suggest you to go through the assignment problems, both which you are doing currently after every week, and also the questions that I discuss here, and also these PPTs also will be available to you. So before your exams, go through all these questions, because your exams mostly will be based on these questions or these type of questions. So it will be helpful for you. Okay. Yeah, the answer is C. Basically, Sanger only determined that this sequence, but this was the protein sequence, not sorry, amino acid sequence, not the DNA sequence, and it was done way back in 1951. So, yeah, it's one of the major milestones. Okay. Okay, let's see, do we have any answers? Okay. Okay, so Edman degradation, I think there is a spelling mistake here. This will be Edman, not Edman. Anyway, so yeah, so this is something that we have not discussed in today's session, but this has been discussed in the week. So Edman degradation is a technique where you sequence amino acids in a peptide sequence, right? So there you do this. There basically. You cleave these amino acid sequences use from the N terminal, and then you one at a time, and then you identify what amino acids were there, and there also cyclization also occurs, and it is through trifluoroacetic acid. So basically what happens is the phenyl isothianate reacts with the uncharged N terminal group, and in the cleavage step the modified terminal amino acid is split off as cyclic derivative without disrupting the rest of the peptide bonds, and it uses TFA. So I think a lot of people said C, if I was correct. Yeah, so that's correct. Okay. Yeah, so these basic things, these kind of questions can come in exams also. So please read up about these things. Okay, I can see different answers. Okay, I'll give another 30 seconds. Okay, there is a simple way to remember this. So generally, so if you have a DNA sequence like this, what you will do is first you will have a DNA sequence of, like, so first you remove the A, you have a sequence, then you remove a T, then you have a sequence G, C, and like that. So you remove one base, you have a sequence like that. So generally, if your length of the base is N, so you have uh, uh, lengths of DNA fragments for Maxim Gilbert starting from 1 to n minus 1. So if this length was 8 base pairs, so the unique fragments produced will be n minus 1, which will be 7. So the answer would be 7. I think there was a lot of 6 and 8. I don't know what is the concept you're using for that, but the idea is that if n is the length of the sequence, Maxim Gilbert will produce n minus 1 type of fragments, unique fragments. So here the answer would be 7. Okay, moving on. Yeah, again, this is something that if you know, you know. So, yeah, bacteriophage M2 protein coding gene was the first completely sequenced gene, and it was done in 1972 before Sanger, so that's also there. So, yeah, the answer is option 1, bacteriophage M2 protein coding gene. Okay, moving on. Do you have any answers? Or, yeah. Yeah, it's not all. The DDNTPs were not replaced by fluorescent DDNTPs. The radioactivity was still used. Sanger does not use fluorescent DDNTPs. Fluorescent signals are used by pyrosequencing and also Illumina and other sequencing technologies. Sanger does not use fluorescence. It still uses radioactive DDNTP. But yeah, slab gel was replaced by capillary tubing, for which actually the throughput increased, and basically excuse me, previously you had manual slab gels. Instead of that, you used automated capillary electrophoresis, so which increased the throughput and reading of the sequences. So actually the first and the third points are connected, but radioactive DDNTPs are still used, not fluorescent. You can use fluorescent. It's not a big deal, but that's not part of the actual Sanger sequencing. In Sanger, radioactive labeling is only used. Okay, so yeah, just be careful with these kind of questions. Okay, I can see a lot of nuns and okay, I think this is for older. Couple of D's and okay. Wait for 30 more seconds. Okay, the answer is actually none of the above. So throughput would be the total amount of DNA that you are generating in a day, right? So that would be product of runs times product of reads per run, and then product of base pairs per read. So if you do, the throughput will be around 1.05 megabase per day, which is not listed here. So which will be none of the above. So it's basically, if you have to multiply all the terms like runs per day, reads per run, and base pair per read. So if you multiply that, you get the whole throughput. Okay, moving on. Okay, this is a straightforward question. Or somebody answered 2002. That is definitely not correct, because I think that you are confusing with the Human Genome Project. It is basically 2023 from the Telomere to Telomere Consortium. So I would just like to yeah. So this is the 2022 paper through which was published in Science, through which they kind of sequence the whole genome. First gapless human genome is the T2T. So other versions of this genome is basically GRCS 38, GRCS 37, older versions. Those had some gaps in some centromeric regions, some repetitive regions. It was not very confident, and so on. T2T is basically one of the most complete genomes that we have, and we actually have more than 99% sequence covered. So still not 100%, but very close to 100%. So technically the 100% written here is not correct, but it is very, very close to 100%. So yeah. Yeah, this is a straightforward one again. Yeah, all of this. So ENCODE is basically it stands for Encyclopedia of DNA Elements. What it was initially made for was basically, since we know that like more than 90% of the DNA does not have any protein coding sequences, right? So there are intronic sequences, intergenic sequences, and other sequences which do not contribute to protein coding. So initially it was thought that they were junk DNA, they do not have any effect on the overall organism, but that was not true. So this particular consortia, what they did was they tried to find what these extra junk DNA actually meant. So they found a lot of things. In intronic intergenic regions, there can be regulatory elements, there can be other genic regions, pseudogenes, and so on and so forth. So basically ENCODE kind of encompasses all of the major elements of DNA. So this is the first ENCODE paper published in Nature in 2012. So yeah, mainly they focused on regulatory elements, but actually everything is almost present. So if you want to have a look later, please go through this. Okay, C, ABC, EB and AC. Okay, okay. Yeah, the answer is the first and the second. So, as I said, throughput increased because of you having those flow cells and you being able to parallelly sequence millions of reads of DNA together. The cost effectiveness also, the cost decreased, so those two are fine. Read length has not increased. In Sanger sequencing also you could do around 5 to 700 base pairs. In Illumina as well you can do the similar thing, about 5 to 700 base pairs. Error rate also, it's almost similar because the uh, the concept of sequencing by synthesis is almost similar. So chemically there is not much difference. It's just that how you are perceiving the signal. So in Illumina you have fluorescence, in Sanger you have radioactivity and so on, but like the general chemistry remains the same. So the error rate and the read length did not was not bettered, but the throughput and the cost effectiveness definitely was better. So yeah, A and B are the options. Okay, read length definitely did not increase. You can make an argument that the error rate probably decreased a little bit, but that too not substantially. But read length definitely did not increase. B and D seems to be the same. Oh, yeah, you're correct. Yeah, this is a typo. Yeah, you're correct. B and D are the same. Okay, anyway, the answer is basically B and D. Yeah, so as the sequencing cycle progress in, say for example in Illumina, phasing or pre-phasing issues and the signal decay cause the base quality to drop, as we were discussing. That sequencing by synthesis, the issue is that the base quality will drop as the length of the read increases. So if it is dropping, then the function will decrease, so the first derivative will be 0, and the degradation will usually accelerate when the run gets longer, and even the second derivative will also be less than 0, because the downward trajectory increases more with the accelerates more with the increase in the length. So either B or D, because they look like the same option here. So yeah, B and D both are correct. Okay, sorry for that, I'll remember this probably. Yeah, let's see. Okay, moving on. and I can see a couple of A, D, and A, B. Okay, the answers are like A and D. Ion torrent and pyrosequencing came out of about in the second time. Uh, uh, around the same time, around early 2000s. So both of them are like higher throughput than Sanger, and both of them actually, but lower than Illumina. But actually, all Illumina's pyro sequencing, ion torrent, all of them are actually second generation sequencing. The first generation is only Maxim Gilbert and Sanger. And 10x and high C are basically not sequencing experiments. High C will tell you, high C is basically chromosome capture configuration. So what happens is like inside the cell, as you know about DNA packing, so the DNA will be wrapped around these histone octamers, and they form these stable 3D structures inside the cell, where one part of the DNA will be in contact with the other part, even that those DNA parts are like, say for example, millions of base pairs away. Then also they can be physically together because of the packing of the DNA that can happen. So high C 10x genomics, these are basically used to capture this long range information, and it's not sequencing itself. Pyro sequencing and ion torrent are your you can say the first second generation sequencing techniques after Sanger and Gilbert, and Illumina is one of the last second generation sequencing techniques or next generation sequencing techniques. So yeah, A and D are the correct options. Yeah, okay. Yeah, this also, if you know, you know. If you don't, you don't. Okay. Yeah, I'm not going to spend much more time here. It's tRNA. It was sequenced in 1965. So, yeah. And it was particularly the alanine transfer RNA. So, okay. Okay, one answer. We'll wait for another minute. Three point two is correct. Uh, I think 400 is a little bit off, I think. Yeah, 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 that's correct. Yeah, yeah. So biotin is 244 Daltons. So 2 biotins is 488, and if you take 660 Daltons as around 1 DNA base pair, so 20 base pair DNA would be around 13,200, and then you add 488 to it. Say 30,688. Generally, like molecular weights like biotin and all would be given in exams, but still I would urge you to know the molecular weight of biotin, because questions like this can come, and also the molecular weight of a DNA molecule as well. So the answer is whatever, I think option A, yeah, 13,688. Okay. Okay. Yeah. Okay, I'll wait for a couple more minutes. Okay, no answers yet. Okay, I'll discuss. One thing to keep in mind is this part. I've taken the screenshot from the content itself, that the genome assembly size will be the sum of the contact lengths, right? There is no gap involved, right? If you just sum of the contact lengths, it will give you the genome assembly size. So the gap that is given is kind of to confuse you. So you do not need the gap information here. So here it is given that longest contact is 131.072 MB, and the shortest is 2 KB, and it's in geometric progression. If you just forget about the gap size, the minimum contact length is 2 KB, the maximum is 131 MB, and they are in geometric progression. So if that is given, I don't know if you can read the handwriting or not, but yeah. So the sum of a GP is given by this formula, a times r to the power n minus 1 by r minus 1, and the nth term is given by an equal to a1 times r to the power n minus 1. So if you just substitute the longest and the shortest here, so the longest will be 131.072 kb. So if you substitute that, and the shortest one, a1, will be your shortest one, so the start of the geometric progression will be your a1, so that will be 2. So 2 times r to the power n minus 1. So here the r considered is 4. So the generally yeah, so generally we consider, I think, yeah, just a second. Okay. Yeah, the r is 4. I think that is not given in the question. You have to consider the r equal to 4, the common ratio, right? I think that is not given here. Okay, this question seems incomplete. So the thing is, when you're calculating, so this is the equation, right? So an equal to a1 times r to the power n minus 1. R is your common ratio, a1 is your first term, an is your last term, and n is the total number of terms. So here, to calculate n, you need the r. The r is assumed to be 4, but it is not given in the question. So I think this question is a little bit off. I'll confirm with other people, but I think yeah, r should be given earlier. So if you have r, you can calculate n would come as 9, and then you can calculate the total sum. So the sum will be a1 would be your 2, r would be 4, n would be 9, minus 1 by 4, 4 minus 1, but this r needs to be given. Yeah, this question is a little bit problematic, I think. Yeah, but the main thing is, like, the question was given to test out the fact that there is no gaps in contics. So if you just sum up the contic length, that will be your genome assembly size. You do not need to care about the gaps. So yeah, so yeah, the thing is, the value of r is missing from here. It's assumed to be 4 here. It's given to be it is shown to be assumed to be 4 here, but it is not given in the question. I will look into it. I'll just mark it down and I'll look into it. Have you guys done the first assignment of your course? Like, is the first week assignment done? Did you have this question or something related to this? Okay. Okay. I'll take a look into this. Yeah, but yeah, the main concept, do not forget this thing. Genome assembly size equal to sum of contic lengths. There is no gap involved here. So you just need to find the total sum of the contic lengths. But yeah, r has to be given. It is not given here. Okay, I'll check this one out. Okay, moving on. answers yet. Okay, let me discuss. Okay, so the idea was that you had the highest length of your was 131,072 kb and your lowest one was 2 kb. So if you do use geometric progression, you can find like what are the contigs present. So you start from 2, 8, 32, 28, 512, 2048, so on and so forth. So if you add all the things up, so you get the total length as 174,762, which is basically, I think, this one, the second option that we got here. So if you add all of them up, that's your genome size, right? So if you add all the contigs up, that's your assembly size. Now you calculate N50. N50 is the 50% threshold of the total genome size. So if your total genome size is 174,762 kb, 50% of it would be around 87.3 kb. So now what you do is you sum the contic lengths from the largest to the smallest, and you see at which position you are crossing this 50% 50% criteria. So from the largest to the smallest, the first contig is only 131,000 kb. So that automatically is crossing the 50% threshold. So your N50 would be the first contig length, which is around 131,072 kb. Similarly, for N90, it will be 90% of the total contig length, total genome length. That would be 157,285 kb. And if you again do from largest to smallest, if you add the second one only, it will become it will cross N90. So if you add these two up, the N90 would be 163,840 kb. And if you calculate the difference between N50 and N90, it is around 100,000 kb, around, I think, 98,304 kb, which is I think the first 98,304 will be your first option. So I hope you got the concept of N50. N50 is basically what is your total genome size, which will be the sum of all the contigs. What is the 50% length of that genome size, whatever you get. Then you see, adding from the largest to the smallest contig size, what is the total contig length that is actually crossing your 50% threshold. So in the first case, your first contig length only crosses the 50,000 threshold, so that contig length is your N50. And for N90, you had to add the second contig length also, which and then it crosses the 90% threshold, which is which adds up to around 163,000 kb. And if you take a difference of this, it is around 98,304. So option A will be the correct option. Okay. Anybody has any questions regarding this? Yes, I'm going a little bit faster, right? If you do not understand anything, please stop and ask. Okay. Okay, moving on. Sorry, I think I got disconnected. Sorry for that. Yeah. So I missed if there was any. Okay, I cannot see the chats anymore. Okay. Okay, so this is what I was discussing. So the question is, which of the following can be used to make Y chromosome probe during Y chromosome extraction process? The answer is streptavidin. Sorry, the answer is B7 and FITC. Streptavidin is also actually used. Streptavidin is also very affinited with respect to biotin, but FITC and vitamin B7 are specifically used for actually probe detection with respect to Y chromosome. But streptavidin, remember the name also, because this is also very widely used uh, for its affinity towards biotin. So all of this, vitamin B7 is already biotin, which is already used. FITC is also used for fluorescent tags, but streptavidin, what happens is it's very it's very high affinity towards biotin itself. So if you want to pull out the uh, the probe from like a mixture of other probes, and if you have biotin attached to the probe, streptavidin is actually a very good solution for that. But yeah, in this case it's FITC and vitamin B7. Okay. Okay, moving on. I'm still there, right? Yeah. Okay, the question is, which of the following sequencing technique involves restriction digestion of DNA? The answer is optical map assisted genome assembly. The restriction enzymes are only used where you want to cleave a DNA molecule at very particular location with a very particular sequence, right? So cleaving is also done in other situations. Say for example, in Maxim Gilbert also you do cleaving, but that's a chemical based cleaving. So you have a chemical tag there, and then you're cleaving. But in this case, it is based on sequence based cleaving. So they use these restriction enzymes. We will discuss optical genome assembly a little bit in further lectures, but yeah, for now, yeah, the answer is optical map assisted genome assembly. But in remember, in Maxim Gilbert you do not use restriction enzymes. There you use chemical tags, and using those tags you degrade cleave the DNA, right? Okay. Okay, yeah, this is the final question. So before you answer this question, you should know what coverage is, right? So the thing is, when you have a whole DNA segment and you are sequencing it, you do not sequence this whole segment once. What you do is you break it down into smaller segments, into overlapping sequences, and then you sequence one position of the genome multiple times. So how many times one particular position of the genome has been sequenced on average, that is the coverage. So 49x means on an average, at every genomic location, there are 49 sequences, reads basically, that have been sequenced to support that base at that position. That is what coverage means, right? So if you have a coverage of 49x, meaning on an average you're sequencing each base 49 times on an average. In some cases, in some regions of the genome it will be higher, in some cases of the genome it will be lower, but on an average it's 49x. So that is what coverage means. So let me first see if anybody has given an answer. Okay, okay. Yeah, so your coverage will be number of reads times the read length divided by the genome size. So basically, if you do genome size times the coverage, that will be the total number of reads times the read length. So that is the formula, and when you plug in all the values, so C will be 49, your genome length is 3.2 billion, so 3.2 times 10 to the power 10 to the power 9, and the read length on an average is 200. So if you solve for this, n will come as 7.84 times 10 to the power 8. So I think some people actually told B, I think, yeah. So that's the correct option. So that's correct. Okay, so that is all from my side for today's session. If you have any questions, you can ask, or else we'll close the session and we'll see you next week. Please ask if you have any questions. Okay, question 7, just let me go back. Okay. From my understanding, it was not like Anjika. So I'm not okay. I will check this, but from my understanding, radioactive labeling is still used. It is fluorescence labeling is probably not used for sinus sequencing now also, but I will check this. But from my understanding, fluorescent labeling was not used. So yeah, I might be wrong, but I'll double check this for sure. I'll discuss this question, and if I get the solution to the other question in the early next week's lecture, I'll come back to you, Anjika. Yeah. 16, 17, 20. Yeah, I do not like. I'm not sure if there are any sources like that. If you practice these questions and also the assignment questions, I think that should be fine. I'm not sure if there are. Meaning, like, if you Google it, you'll definitely find. But off the top of my head, I do not have any book in mind. Yeah, no. Yeah, I'm not sure about that. Aditya.
Mr. Majumdar, I want to ask one question.
Yeah, sure.
Thank you for your nice presentation. If we practice the assignment questions and the questions you have provided here and discussed the answers, so is it enough to pass through the exam?
Yeah, my understanding would be it is enough. You should have a good understanding on the concepts that are discussed. Question wise, from what I understand, is the questions that are asked in the examinations are not very difficult. They are of standard level. It's not like that you cannot answer those questions at all if you if you do not vehemently practice those or something like that. But the questions would be more or less based on these kind of questions only. So if you practice the assignment questions, there are, I think, also some practice questions are also there in the module, if I'm not wrong. And also these questions that I'm discussing are previous year questions. So the final paper is based mostly on this. Some questions may come obviously outside what we are discussing, but for that, if you just follow the lectures and you have your concepts clear, you will be able to answer. It's not going to be that difficult.
Okay, thanks a lot.
Yeah.
Hello, sir. Good evening. Can you please help that like we are using various tools, no, in NGS, So how can we decide and what are the sources through which we can get the various kind of tools used for similar kind of task?
Tools as in what kind of tools? Are you talking about the sequencing experiments itself, like how you are generating the sequence, or like after generating the sequence, how you are analyzing the data? Which part are you talking about?
Analyzing parts.
Okay, I will discuss in quite a bit about the analysis, data analysis part, because that is what I do. I am a bioinformatician. I'm not a white lab person. I generally do data analysis only, with specifically with genomics. So I am going to discuss those things in detail in the coming lectures. There are a lot of so so what happens is there are set pipelines in place. Right? Nowadays we have pipelines in place where you just input the data and it'll do all the processing and it'll give you the output. So there are a lot of pipelines. So Illumina has something called Dragon, DRAGEN. So that's a whole module pipeline. It takes the raw FASTQ data and then processes everything and gives you an output on terms of like what are pathogenic variations present and what are the different haplotypes present and so on and so forth. So it currently the technologies we have gives you the whole thing. Now what is happening inside that is important, like what steps are getting followed, what kind of algorithms are getting used. I will discuss that definitely for the genomics part, of course. For transcriptomics also I'll discuss a little bit. After then, if you have any particular things to learn, I can definitely give you some sources. On the all overall thing, I cannot, like I do not have anything in mind to tell you, like if you follow this particular paper or this particular like article, you'll be fine. It's like combination of different things. After I discuss that, then you let me know what kind of things you would like to learn more, or like more you would want more understanding in. Then I can probably help you with some sources. Currently it's very vague. If I give you something, it'll be very end to end thing, and it'll be harder for you to understand. So after I explain, maybe you can then take a look, and then you let me know.
Okay,
That will
okay,
be fine.
thank you.
Yeah. Let me see if I have anything. Yeah, if you just want to look at the state of the art technology, so this is one paper which came out in I think 2023 or 2024. So this is basically what I was talking about, Dragon. So this is a Illumina proprietary pipeline that they use to analyze genomic data. So they start from the very basic. They start from FASTQs, then they do alignment, then they do variant calling and everything. But this tool is not open source. Illumina charges, uh, like you have to pay to use this tool. So yeah, it's not free. But you can take a look at the paper if you want to have an understanding. But I would suggest like you go through this class, and then probably like after when I discuss these things, then probably it'll help you understand better. Okay, anything else? Okay, and one more request is please do not call me sir, you can call me Shomo, and this is my email ID. If you have any questions, or if you have any thoughts, or if you are uh, like interested in genomics, you want to learn something more or anything, like you can email me here. I'll definitely get back to you. Okay, thank you guys for joining. We'll see you next week. Thank you.